This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 50, recorded on January 24th, 2013. Hello, everyone. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent. Good to be with you this afternoon.
Now, it seems like a long time, but it's only been 2 weeks. Is that right?
That's right. But, you know, January's almost over. 12th of the year gone.
Amazing. Yeah, January is almost over. Also joining us today from San Diego State University, Stan Malloy. Hey, Stan.
Hi, Vincent and Michael. Good to hear you guys' voices.
Haven't had you on the show for a while. How have you been?
I thought you were just upset with me or something.
No, not at all. We try to leave you alone.
Things have been going well.
Very good.
Do we get to talk about the weather? I can complain about how miserable it is in San Diego today.
How miserable is it?
Well, it's cloudy. The high is only 72, and it's going to get down to 55 tonight.
55. Well, here we've had a cold spell here in New York, in the New York area. Currently, it is -6 Celsius.
Ooh.
It gets down to -10, 11 at night. And this is what's in store for the rest of the week, the weekend. It's been pretty cold. How about you, Michael? You're probably warmer than that, right?
Yeah, we got the Arctic blast like the rest of the East Coast of the United States, but our definition of Arctic isn't even close. It's, in the 50s. So it's not too bad, and there's nothing much to complain about. The sun is out, and it's a beautiful day. So we're very thankful. And this morning I heard that the poor folks in Staten Island are still without heat, many of them from Hurricane Sandy. So consequently, we certainly feel for those individuals, and I wish I could send them some of this 50-degree weather so they could at least, do a little bit better.
I have a colleague here who's retiring this year, and maybe, Stanley, you know him, Dave Figursky?
Yes.
Works on AA, the bacteria that I can't pronounce.
Actinobacillus actinomycomatans?
Yeah.
Right.
But it's changed its name again. It's now something else.
Really?
Right.
So he's retiring in September and moving to Taos, New Mexico. And he said now he's having second thoughts because it's about 10 degrees colder in the winter than it is even here, and he doesn't like it here at all. So I said, you got to go to San Diego where it's 70 and sunny all the time.
Right.
He can go visit you and Elio. It'd be fun.
Absolutely. Send him out.
Yeah. So San Diego, I'm sorry, you guys cannot complain about the weather.
Well,
hopefully Elio will join us later, but we're going to start talking today. And the reason we have Stan here, well, Stan's always welcome, of course, but he's a busy guy. You're a dean, right, Stan?
That's right.
So you can't be bothered with these little podcasts here, but we thought we'd bring him on to talk about Carl Woese, who just passed not too long ago. We had a little conversation, and we decided that you would be a good person to talk about it. And I understand that Michael knew him or his career as well. So that's what we're going to do. And hopefully Elio can join us because, as you say, Stan, he would also have a lot to say about this.
Absolutely.
I don't even know where Carl is from, Stanley. So do you know, can you fill in the early years?
Yeah. So I actually, let me give a perspective here. I've known Carl for a long time because I spent 18 years of my career working at the University of Illinois in Urbana-Champaign, which is where Carl spent the vast majority of his career. And so he was right down the hall.
Same department as you?
The same department. And we got to know each other. I'll tell that story a little later, but we got to know each other, and often in the morning he'd drop by. He and I were the earliest people to get into the building, and he'd drop by and would just talk about some problem that happened to be on his brain at the time. So I had the wonderful opportunity to talk to him about a large number of different things.
Wow, you have a really a good insight into what he was doing then, right?
I never worked for him, but I talked to him a lot, and we even co-authored a paper together once. So
nice.
But so anyway, he was from Syracuse, New York, and he was born in the Depression era. And growing up in the Depression, he really was focused on very practical things. So he went off to Amherst College in Massachusetts and got a bachelor's degree in physics. From there, he went on to Yale, and he got a PhD in biophysics. And decided at that time that he thought he'd like to go into medicine. So he went to medical school for 2 years, and he hated it. And so after 2 years, he bailed from medical school. He went back to his PhD advisor's lab at Yale and began doing a postdoc. And his postdoctoral research was focused on, at the time, physicists were really interested in activation of biological of viruses, of bacteria, of different spores, you name it, using radiation. And so this is what he got involved in. And his particular area was on spores of Bacillus subtilis. So he was interested in the germination of spores and how you could inhibit the germination. And that led him, this was really a critical time of the basic blossoming of molecular biology, of our understanding of central dogma.
Was this in the 50s now that you're talking about?
Yeah. So it was in the mid-50s to late 50s, and he became really interested in the genetic code and in proteins. And in particular, he was interested in information, though. So a lot of people were interested in just in mechanism, but Carl was interested in, how the heck did these things evolve? Right? How does information evolve? And so it was that early work that I think set the stage for his whole later career. So he had a couple of positions working at General Electric, but then in 1964 he moved to the University of Illinois in Urbana-Champaign, and at the time, this was really a hotbed of people working in molecular biology coming in and thinking about things. And so Saul Spiegelman was there. Mm-hmm. Saul Spiegelman is really credited with the development of the concept of hybridization. So this is an enormously impactful way of looking at relationship of molecules. And for a while, Watson was there. Luria was there. I mean, it was really a place where a lot of people came. And so he started there, and he started working on this problem of trying to understand the genetic code, and that led him to working on ribosomes. And he got this idea that ribosomal RNAs may provide the key for understanding evolution, for comparing organisms. Right? So there are some people who say that this was he was the first one to think about this, but in fact, I think that wasn't true. I mean, so Crick and Pauling before had proposed similar things, but their idea was to look at protein sequences. And in fact, some groups had begun to do sequence comparisons of cytochrome C. And those studies were really important for understanding a part of evolution. But because cytochrome C is not in all organisms, they only saw a little bit of the biological world. So Carl said, well, all organisms have ribosomal RNA, so let's sequence the ribosomal RNA to do these comparisons.
So that was his idea to do the sequence, is that right?
That was his idea.
Okay.
And it was a horrendous job, right? So in those days, to do this, you would take a millicurie or two of DNA. Note that I said milli, not micro, a millicurie or two of P32, and you'd label up the nucleic acids in an organism. You'd purify the nucleic acids, and then you would take the RNA, you'd treat it with a number of nucleases, you'd separate them on 2D gels, pick a spot, you treat it with more nucleases so that you could get the sequence of all of these different bands. And you do the autoradiography of them, and then you'd sit there and think, how does this all fit together, right? And so Carl had a series of people that were phenomenal experimentalists in his lab that could do this work. And Carl, there's this classic picture of Carl sitting at the lab bench looking at an autoradiogram with his feet up on the bench. And that's classic Carl. He sat there and stared at these sequences all day, every day. And he loved it. He never tired of it, right? But so they started off looking 5S ribosomal RNA, and the problem was that sequence was too short. You couldn't look at organisms really broadly. So he switched to using 16S ribosomal RNA. So this is about 1,500 base pairs. And it was a great sequence to look at because parts of it are very conserved and parts of it are highly variable. So the conserved parts allow you to look at organisms that are really far apart, and the variable parts let you to see differences between organisms that are closer related. Right? So this proved to really be, this is really Carl Woese's genius, was recognizing that this would allow you to understand the relationship between organisms.
So this was done by labeling the RNA of the ribosome. So he added P32 to bacteria, right? And then purifying the ribosomes and then breaking up the nucleic acids. Is that right?
That's right.
Okay, so that you're working with a lot of radioactivity.
A lot of well, and actually, the way that you had to cool these gels in those days was you'd use some organic chemicals in the bath, and the gels themselves were enormous gels. And so not only were you using this high level of radiation, but you're using potentially flammable things in your water baths, and you're running this at currents that would readily electrocute somebody. So really safe work. But it was really important, right? And so they did this work with a whole bunch of different bacteria, and then they did it with a whole bunch of eukaryotes, and they compared the bacteria, and they all clustered together in terms of similarity of the sequence of the RNA, and they clustered the eukaryotes, and they were different from the bacteria, but they were similar to each other in the ribosomal RNA. And so this led to the idea that, in fact, the ribosomal RNA was a way of distinguishing these two really important groups of organisms, the bacteria and the eukaryotes. So,
now, at this time, were you there with him at Illinois, or this happened before you got there?
This all happened before I got there. This was happening when I was an undergraduate, actually. And in fact, at the time he was doing this, the people who paid attention to it were mostly evolutionary biologists. At the stage that I've just talked about now, I don't think this had really hit the mainstream in a big way, right? But then there was something really powerful that happened, and this is one of these great serendipity stories. So down the hallway from Carl Woese was the lab of Ralph Wolff. And Ralph Wolff's lab worked on methanogenic bacteria. And so at the time, very few people in the world worked on methanogens. They were really strict anaerobes. They were very hard to grow. You had to have a green thumb, basically, to grow these things.
Where did he get them from?
So they got these from a lot of places. So the methanogens are present in rumen of animals. They're present in, out in swamps, in a number of different places. I mean, people knew about the concept of methanogenic bacteria for many, many years because in swamps, you release the swamp gas that can catch on fire by methanogenic bacteria. But so anyway, in the process of his lab studying these bacteria, Ralph was a premier bacterial physiologist and biochemist. And they discovered really unusual coenzymes. and they discovered really unusual biochemical pathways. And this really bothered him because there was this idea that we were all taught to believe about this unity of biochemistry. Remember, once upon a time, Jacob said, E. coli is the same as an elephant, only more so, right? I have that backwards. An elephant is the same as E. coli, only more so. That makes a little more sense.
Yeah, right.
But so anyway, so Ralph has these really weird organisms. They don't seem to conform to any of the other organisms that are known by anyone in the world. And so he bumps into Carl Woese, and Carl says, oh, why don't you let us look at the RNA of these? And so a graduate student in Ralph's lab and a graduate student in Carl Woese's lab, they got together and Falk provided the organisms and Fox sequenced them. Woese came back to Ralph and said, that didn't work. Something was screwed up. Tried it again, and they tried it again. And Carl came back to him and said, the organisms you have, this isn't bacteria. So the idea that there was this third group, third domain of life that was as different from eukaryotes as they were from bacteria, I mean, this was really a revolutionary discovery. It destroyed our whole conceptualization of the dichotomy of life. And so it made it to the New York Times, and it really pissed off a lot of biologists who were really ingrained in an old way of thinking, right?
And I was at Indiana at the time, and one of the microbiologists, who was really taken aback with this. I still remember when this day happened, and it was in the New York Times, and they went and looked at the paper. This was Howard Guess, the great microbiologist who spent much of his career describing new photosynthetic microbes. And Howard was aghast. And Howard, of course, in the 80s began working on stromatolites, looking for ancient bacteria. And so this was an extremely controversial topic. And I remember he and my advisor, Walt Konecka, having this discussion. And as a young, impressionable graduate student sitting in the corner watching these two men argue, and it was a full-blown argument about the validity of whether or not there could indeed be a third kingdom, because Dr. Guest and Dr. Konecka were these tremendous bacterial physiologists who well understood anaerobes, and methanogenic bacteria were ingrained in the microbial world, and they must be bacteria because they were following the old adages, if it walks like a duck and talks like a duck, it must be a duck. But what Woese, and this was in the early days when they were looking at 5S ribosomal RNA sequences, that's what Woese initially looked at, something that had less information knowledge in it than the 16S that many of us use today to identify bacteria. And so it was extremely controversial. So as a young and impressionable graduate student, and this wasn't assigned to us to read, you immediately, this was the days before the journals were on your laptop computer, because laptop computers were but a mere dream of graduate students at the time. We went down to the stacks and went looking for the journal that this was published in to see what all the fuss was about, and to begin to wrap your head around this concept about a molecule that could actually tell you how old something was, because the name they gave the kingdom, Archaea or Archaebacteria, implied that it was ancient. So that was controversial. And I think that's what made it so exciting for Howard Guest is, he was wondering whether they should be called ancient because are they just simply different? And so, Stanley, what was going on in Illinois at the time?
Well, so again, this is before I got there. This is in 1977, and I got there in 1984. So a lot of this initial stuff had happened before I arrived. So even the name, right? So they're now called Archaea. They were called archaebacteria, and that was because people could not accept the idea that they weren't bacteria, that both of them are prokaryotes, that is, they both lack a nucleus, so they have to be related to each other, right? And you have to Guest and all the other premier microbiologists at that time, they said, they basically gave up on having a true phylogenetic approach to organizing bacteria and other prokaryotes, if you will, that is bacteria and archaea, because they said, it's too messy. There's no characteristic that they all share that we can do this comparison. So they had all kinds of real funky ways to say, well, if they share this many metabolic traits versus that many metabolic traits, then we'll call them this versus that. And along comes Carl Woese and his group, and all of a sudden, there is a phylogenetics that makes sense, and it tells an evolutionary story, right?
And you can do math with it.
You could do math with it. On your website, you have posted one of the papers by them. This is a paper by Woese and Fox in PNAS in 1977. It's called Phylogenetic Structure of the Prokaryotic Domain, the Primary Kingdoms. Okay, this paper is elegant. It is one of those kinds of papers that is a pleasure to read, not only because the insights were so impressive, but because the writing is so clean and crisp and nice. Now, this whole paper that's laying out this idea of the 3 domains of life has one table. One table. It doesn't have 14 figures and 5 tables, right? It has one table of data. And that table of data is basically just a comparison of how closely related these ribosomal RNA sequences are. But when you look at this table, the numbers hit you in the face. Bacteria grouped together. Eukaryotes grouped together. Archaea grouped together. And so this is a great example, not only of a really important discovery, but a way that you can lay out something that is elegant, understandable, and simple. So,
and it's still worth reading today. This paper is still, it should be primary reading for any freshman biology class, because it really lays out the differences and really, it could almost teach a freshman biology major that you really need to begin to understand various aspects of math and various aspects of statistics in order to really make these data interpretable. It's just incredible.
Stanley, they mention in this paper all the phenotypic differences between archaea and bacteria, which had been known before the ribosomal sequence was determined. Didn't this hint that these were really very different?
It hinted, but sometimes you can have all the hints in the world, and it's just one piece of data that throws you over the edge. And in fact, if it had not been for all that biochemical data showing the differences, I think it would have been even harder for people to accept this idea of the 3 domains, right?
And of course, one of the major differences between the bacteria and the archaea is that the common protein synthetic antibiotics that we use to control bacteria don't work in the archaea.
Yeah.
Because the ribosome is different, but yet it's similar enough that we can get information from the same molecule.
Yeah. I mean, this is really, I think, the real crux of the genius of Carl Woese. That is, early in his career, he became fascinated with understanding this idea of evolution and how you can track evolution and how it ties into what was called central dogma at the time. That is, the very central processes of DNA to RNA to protein. And because he was so focused on that, it allowed him to develop a technique that nobody else had that could really probe this process in detail. And then he really stuck with it and collected a volume of really detailed data, a tremendous amount of work that went into the early studies here. And then the serendipity kicks in. He has this bump in with Ralph Wolff and says, well, let's try looking at those organisms. And that really had the big shift that turned this from something where a lot of the world could say, oh yeah, they can look at these sequences all they want. This is just about bacteria. Who cares, right? To something that all of a sudden it reorganizes the way we think about the world to an extent that it did not take long before this percolated into textbooks, right?
How long was that?
Yeah, it was less than 10 years, I think.
Yeah.
I have a hard time putting that in place, but I know that when I was an assistant professor, which was less than 10 years from then, this was a standard part of lectures in general microbiology class.
So by then it was universally accepted, right?
There's still some people who doubt that the Earth is round, right? I mean,
yeah.
Yes. Yes, but for the most part, right? For all the reasonable microbiologists accepted that this was the case, right?
So one of the people who hated this idea was a really preeminent evolutionary biologist, Ernest Mayr. And he hated this idea because he'd spent his whole life studying eukaryotes, and his career had been based on comparison of structural features of animals, and so he just undermined him in a way that was very bothersome. And he had a tremendous amount of clout, right? A very big guy. And there were a lot of other people who kept arguing about this and whining about it. But I think that it was so clear to people who knew molecular biology that it rapidly at this time, molecular biology was permeating every different discipline in biology. And I think that the younger generation said, this just makes sense, right?
Yeah. So when did other Archaea become discovered and added to this kingdom? Because here in this paper, it's all methanobacteria, right?
So this is the other thing that some people said. They said, oh, you're just looking at these weird organisms that exist in extreme environments.
Yeah.
This isn't really a major part of the biological world. And so that was one of the real early arguments against this. The change came about because of some of Carl's students that began to take this and to think about the next step, right? And one of the guys who really had a big impact, there are a number of them, but Norm Pace in particular. Norm realized that, look, if you can look at the sequence and identify what the organism is, then we should be able to go out in nature, just isolate the nucleic acid. Initially, they did it by cloning, and then sequence it, and we'll know what's out there in nature, even if we can't grow it in the laboratory.
When I was a graduate student, my mentor had as his lab project, he was working on magnetotactic bacteria. In his side project, because he was having a challenging time figuring out how to be able to cultivate these microbes in the lab, was by that point in time, Norm Pace had been recruited to Indiana. And so I remember he and Norm discussing about how to figure out what this magnetotactic bacterium was, the best way to figure out how to design a medium that would support its growth would be to understand where it came from. And so they began to, and you could isolate enough of these magnetotactic bacteria with a stirring bar. You'd put the stirring bar next to the mud, and the magnetotactic bacteria would swim out, and then you would collect them, and you would be able to isolate enough of the magnetotactic bacteria from which you could then isolate the RNA. And by this point in time, Pace's lab had pioneered the transition from 5S ribosomal RNA sequencing to 16S ribosomal RNA sequencing, moving it from the Maxim Gilbert, now I'm really dating myself, the Maxim Gilbert type sequencing for nucleic acid to the Sanger style of sequencing using deoxyribon. They would go through a step where you could clone it, and then via Sanger sequencing, you could get the sequences much more easily than had been done previously in the past with these really spooky chemicals that Stanley was talking about earlier, where you could literally blow up the lab if you didn't watch yourself carefully. And so it was really Pace and the students that came out of Pace and all of the other of Woese's students that really began to poke around. And then again, serendipity comes into play, because about that same time, the polymerase chain reaction became commonplace because of advances made by Tom Brock, who isolated the DNA polymerase from Thermus aquaticus out of the hot spring of Yellowstone National Park, and then Kerry Mullis, who pioneered using that polymerase in order to amplify DNA quickly. And in the olden times, you could actually do PCR without a thermocycler, just having water baths or heating blocks at various temperatures. And then you, as the graduate student, would literally cycle back and forth between heat blocks in order to do the cycling, doing this sort of technology. And as time progressed, what then happened is computing began to mature, where people began to have biologists began to have ready access to computers. And then the ribosomal database became more commonplace, and people would begin to deposit these 16S ribosomal RNA sequences. And I think really the ribosomal database project that was initially funded, and now it's universally funded, so that you can just deposit sequences, and all of the other tools that the NCBI have pioneered for mining nucleic acid sequences began to mature. And we've really had a rapid expansion of the depositing of sequence into the ribosomal database project. And then the 16S ribosomal RNA sequence effectively became the molecular chronometer that we all know and love today.
So today, even people studying, the microbiome of certain parts of the human body will sequence the 16S RNA to know what's there, right?
Right.
Yes.
And this is done by amplifying the region by PCR and then sequencing the products. So, Stanley, what were the next Archaea? Getting back to that.
Well, so this is a follow-up on what Michael said. So at that period of time, so many microbiologists were beginning to do molecular things that a lot of people started looking down on microbial ecology and environmental microbiology. Said, oh, what can you do? Right? You can't really grow these organisms. You can't identify the organisms. You just kind of out playing in the muck and mire of the ocean, and how important is it? Right? Us molecular biologists like to be really snooty about things like that. But then once you could do this 16S sequencing, you could go out in nature and you could ask who is there. And the amazing thing was that if we try to culture all these things, we only saw about 1% of all the organisms out there, right? So there's all of this 99% out there. They must be doing something really important. And it took a while to fully mature, but this really opened the doors for microbial ecology and environmental microbiology transitioning from being a phenomenological science. Some people are going to kill me for saying this.
Ooh, you're going to get letters. You're going to get letters.
But a lot of it was looking and seeing what happens in different places. But now you could go in, you could quantitate How many different things were there? How much of each one of those things were there? And not only that, because the sequences were associated with certain groups of microorganisms, you could begin to make predictions about what kind of metabolism was happening in those places, right? And so this is a huge breakthrough. So let's go back to the beginning, right? So when Carl started, we didn't have any good way of looking at phylogenetics and a comparative evolution of organisms, right? And then he developed that ability, and now we can go begin to look at what organisms are out in the environment and tie in the whole process of evolution, genetics and metabolism, ecology. This is the full circle of biology, right?
Absolutely.
This is why it totally changed the way that we as microbiologists can look at things. And so when we started doing this, now we go out and we look in the ocean, and we look in people's mouths, and we look in people's intestines, and we see that archaea are everywhere, including environments that aren't extreme environments, right? It's just a third group of organisms that's populated distinct niches around the world. Some of them happen to grow in extreme environments, but some of them grow in our own mouths, right? So what an important change of the worldview for microbiologists.
Sounds to me like he should have had a Nobel Prize.
Many people think so.
What do you think?
Carl won many, many prizes. I mean, he got a MacArthur Award. He got a Crawford Prize. He got a lot of accolades in his life. It ends up that the Nobel Prize really doesn't apply to the area of ecology. Okay, and people initially viewed his work as being something that was important for ecology and evolution and environment, and they didn't think about. I mean, back in 1980, it was impossible to conceptualize the Human Microbiome Project, right? And so,
yeah, we couldn't do it. The Moores at Virginia Tech were so ahead of their time about the microbiology of the human gut that no one could figure out how to do the science. And it's only because of Woese's contributions to understanding being able to identify who's there, that last week in the New England Journal of Medicine, there was an editorial and a paper demonstrating that you could use the human microbiome to effectively treat a chronic illness that was started by an antibiotic that then resulted in the blooming of Clostridium difficile that then resulted in ulcerative colitis in the patient population. And they asked a question: could you cure the patient with, of all things, a stool transplant? And this editorial, and the stool transplant technology is 50 plus years old, but no one really could explain why it worked or to design an ethical clinical trial where they could measure the variables. And something as simple-minded as asking your significant other in your home for a stool and then going to a physician where they whiz it up in a blender and then they give it back to you via enema, that you could literally be cured of a chronic disease in which the only cure before was horrific surgery, where you had to take out the necrotic tissue because if you left C. diff go too long, it'll effectively rot your insides. And this was all made possible by one individual. And so today, you could take Woese's contribution that he began that is now enabling translation of basic science to a bedside application.
So if Carl had lived another 5 years, the types of things that Michael's talked about and many, many others, I'm convinced if he'd lived another 5 years, he would get a Nobel Prize. But, you know, Nobel Prizes are complicated things. And sometimes a person dies before the time is right for it to happen. And in this case, it really reflected the idea that this was evolution and ecology, and it wasn't mainstream things related to medicine or chemistry or physics that I think kept it from being on the docket for the Nobel Prize. I have to say, I think Carl was pretty bitter about that.
Really?
He really thought he deserved a Nobel Prize.
I would agree. I think it's amazing. And the prizes are in part voted on by the previous winners, right? Mm-hmm. And you have, I suppose it would be physiology or medicine that it would be awarded to him. And, you have some virology here. You have a lot of basic science of the cell. You have all sorts of people who should appreciate this, but maybe they didn't. Look, I'm looking at the list. Ralph Steinman, of course, would have loved it, but he was dead.
Well, look at the RNA biologists, Sid Altman and Tom Cech.
Yeah.
The RNA biologists should have appreciated this.
Yeah. Sidney Brenner should appreciate it, right?
Mm-hmm. I really do think if there had been another 5 years, I think it would have happened. Because a tremendous number of people appreciate him. If you look at a number of recent things written on his behalf, and in fact, if you look at his obituary, it made it into all of the major newspapers. I mean, this is somebody who really had an impact.
So the work you've described took place in the 70s, late 70s, and into the early 80s. And what did Carl continue to do until he was still working, I presume, when he died? Is that right?
He still was. He was doing a lot of things. So whenever you make a discovery like this, then you can begin to apply it. So if you want to understand evolution, then you really need to be looking at a lot of different organisms so you can see what happens between these closely related organisms, what happens more distantly related organisms. And somewhere after this had been pretty well accepted, a wrench was thrown in the gears, right? And the wrench that was thrown in the gears really related to whole genome sequencing. By the way, I have to say, Carl Woese got this idea that you could sequence the whole genome of a bacterium. Mm-hmm. And before anybody else had done this, and he had a proposal in to sequence the whole genome of one of the methanogens, a relatively small genome, brought in great people to do it. I was privileged to be part of the group that was having these discussions, put together a grant proposal, sent it off, and it got fired back saying this is impossible. You can't sequence the whole genome of an organism. And then it was not maybe a couple years after that whole genome sequence of organisms was released, and the Human Genome Project started.
And today we sequence bacterial genome between breakfast and lunch.
Mm-hmm.
That's another case where, except for getting funding, he really had this idea that you could do it at a time when it really was kind of a unique and crazy idea, right? And so he wasn't the first, but he definitely was on board with that. But look, so now what happened is once we started sequencing all these organisms and we look at their genomes, we'll say there's an amazing amount of transfer of genetic material between these organisms, horizontal gene transfer, right? And so if genetic material is moving extensively horizontally between organisms, then how can you trust one DNA sequence to tell you about vertical transmission, to tell you about the evolutionary process, right? So when Carl started, we didn't realize there was all this rampant horizontal gene transfer. But after it showed this great stuff, now there was this problem. And so a number of people started arguing, well, you can't really trust 16S ribosomal RNA to tell you about evolution of organisms because we can't build a tree of life. Life is a bush. It's a tumbleweed. It's so complicated and promiscuous and intermersed that you can't sort it out. That's kind of the real extreme argument of this group. But so, all right, now all of a sudden Carl's in a situation where he's got to defend himself once more, right? And the way that they did that was they said, well, look, you get a lot of gene transfer, but when you look at gene transfer, essentially all of it seems to be transfer of things that are accessory genes, not the key functions in central dogma, right? So if the 16S ribosomal sequences provide you a valid tree, you should be able to go to some other key function in translation and say, how do they build a different tree? Is it a similar tree? And so they used a number of things, but I think a really great example are the aminoacyl tRNA synthetases, critical functions for the translation from mRNA into protein. And you build a tree with these things, and it's right on top of the 16S tree, right? So I think that those kinds of studies, and so that also occupied a variety of Carl's time later on, but those studies were really important in terms of nailing the coffin shut about the use of this approach for phylogenomics. Earlier you said, so now this is universally accepted. There are still people out there who will hem and haw about whether you can make interpretations because evolution is a bush, not a tree, right?
And so you can almost make this into one of Elio's famous Talmudic questions. You can ask the question, is the 16S ribosomal RNA molecule and its eukaryote and archaea counterpart the only true molecular chronometer for describing evolution? And are there other molecular chronometers that could be as effective as this one particular molecule? And you really get into discussing, how you begin to define life.
And actually, that's an important point that Michael just brought up, because not only does this allow you to look at the human microbiome, but it allows you to make predictions about how life first evolved on Earth, right? And when he died, just at the end of last year, late December, when he died, Carl was part of a very big astrobiology grant that was focused on answering a number of very key, important questions about evolution, about life on Earth, how we got here.
One of the problems in doing phylogenomics in viruses is that you don't have any ribosomal RNA sequence like this. And since there's so much transfer among viruses, same thing that happens in bacteria and archaea and eukaryotes, you don't have the ribosomal to set you straight, the clock, as you guys said. And this is a big problem that complicates looking at viruses in this way.
Yeah, viruses are so complicated even when you have the complete genome sequence of set of viruses. But, I think that nonetheless, it was the laborious process of going through the RNA sequencing that played an important role in our thinking about doing the comparative analysis to understand phylogenetics of all organisms. And I think that those concepts, although still a bit messy, really spilled over into viruses as well.
So a paper that you provided would be good for our listeners. It was a paper published in PNAS in 2000, July of 2000. It's called Interpreting the Universal Phylogenetic Tree. I think that embodies a lot of what you've been talking about using this information to do phylogenomics.
Right. I think it's a nice beginning point, but as Michael mentioned earlier, going back to and reading the 1977 paper is one of those kinds of things that will really help set thinking in a really effective way. And I think that it's a nice piece of work. And to have had this kind of impact on biology, the number of people who can say that what they did in their life has made it into all of the general biology textbooks, that it's really influenced fields of ecology and evolution and medicine, that it led to a whole series of other studies that would have been possible before that. That's an amazing legacy.
Now, another measure we use of the effectiveness of someone is who they train. Now, are there people that Carl trained that we know about that have continued this and are well known? So I don't know any of that, but maybe you guys would know that.
He trained a number of people who have done really well. You know, Carl didn't train as many people as some folks.
Yeah.
I mean, he didn't have huge groups in his lab, but he trained people who went on to do some really important things. We mentioned Norm Pace earlier.
Mm-hmm.
Right? Mitch Sogan is another one of his progeny. He's continues to do really great stuff in this area. David Stahl.
Yeah, David.
Great stuff. Has had a big influence on this field. And at the University of Illinois, one of his colleagues who was hired there as an assistant professor but became really a close colleague and confidant of Carl is Gary Olsen. And Gary Olsen is a phenomenal bioinformatics guy and thinks really hard about computational approaches to understand evolution. And Gary has also had a tremendous impact on this field. There are others. Those are just a few. But, Carl's people have done really well.
And in a sense, we've all been trained by Carl. You just need to read his papers, and, there's not one practicing microbiologist, whether they be a clinical microbiologist or a microbial ecologist, that doesn't use the concepts and foundation that he built for us.
There's a wonderful webpage over at John Eisen's blog, The Tree of Life, where there's several. He's got one where he collects, he's been collecting a lot of posts on Carl Woese, and he says, my main science hero. He is without a doubt the person who most influenced my career as a scientist. And we'll post a link to that. There's a lot of good stuff there as well. He says the 1977 Woese and Fox paper may be the most important paper ever in microbiology.
Mm-hmm. There are some people who would take exception to that.
Of course, but, it's okay. You can think something is important. It's fine.
That's true.
It sounds to me like it's pretty damn important.
That's right. There's no doubt it was very important.
Now, I was a graduate student in 1977, and I was trying to sequence influenza virus RNA, and we used the same technique. We would label the virus in infected cells with P32, and then we would digest the RNA with T1 ribonuclease, which cuts after G residues, and that's exactly what Carl has in this 77 paper, the sequences of these T1 oligonucleotides. You could purify them on a gel, and then you would sequence them in other ways. You get, 12 or 20 nucleotides of sequence. And it was so tedious and pretty dangerous as well. So I know exactly what they were doing. It's really amazing.
I did a lot of that, labeling cells with milligram quantities of P32. That's probably
millicurie, right?
Yeah, it probably, would I say milligrams? Millicurie. It probably was partly what resulted in somatic mutations and brain damage. That's why I'm a dean today.
Yeah, but you're living a long time, and so that probably helped you as well.
So there was one thing later in Carl's life that he argued very strongly. He said he became anti-Darwin, right? Mm-hmm. This is a really interesting thing, because to be anti-Darwin in many respects makes you pro-creationist. And if you argue against Darwin, even if you're arguing from scientific grounds, the creationists are going to grab it and take any little tidbits they can to put in their documents. The argument, kind of in a nutshell, it's a real long argument, but the basic idea of his concern about Darwin was Darwin really thought about life as being a progression where you move from A to B to C, right? And that's what allowed Darwin to draw his first tree in Origin of Species. The difficulty is we now know that there are a number of other things going on. We know that there's horizontal gene transfer. So that doesn't follow this simple linear relationship. We know that epigenetics is really important in a lot of cases where you may be able to transiently inherit something, and it's not something that's directly encoded in your DNA, right? And there are a number of other processes like this going on that are not the simple linear progression that Darwin proposed. So because of those things, Carl really said, well, Darwin had it wrong, and we need to really have yet another synthesis of evolution that's based on these kinds of sequence comparisons to build what is the idea of the tree of life. This is a situation that Carl and I actually argued about quite a bit. I think I understand the importance of all of those things, but I personally think that Darwin had a real important influence on our thinking. And the concept of natural selection is still a very important concept when thinking about ecology and evolution. So there was a lot that Darwin didn't know. He lived a long time ago. He didn't have the information and the tools we have. I don't think we can expect him to have known those things. But I don't think we should throw out the insights that Darwin had just because of some of these new ideas.
And in fact, it may be the fact that Woese was not trained in the school of Stanier, where you go out to the mud and you select out for the microorganism, or you enrich, and you can really begin to see how things can evolve. And when you look at some of the experiments by Linsky on evolution in a test tube, where you can actually see a pure culture of E. coli change with time through subsequent generations just by adapting to its circumstance, there's a lot of physiology that impacts on the genetic code, and the genetic code isn't as sterile as everyone would like to think it is. You only have to look at what's happened over the last probably 10 to 15 years as we've watched the first genetic code of DNA, and then you begin to look at the second genetic code of these small regulatory RNAs that control expression, and then the third genetic code of methylation, and all of the epigenetic phenomena could actually be the fourth genetic code. And all of these things that are operating on the foundational materials of life, DNA, really are responsible for what ultimately turns out as evolution, because you are the product of your surroundings. And Darwin really elegantly showed it, in everything that he described on those marvelous Galapagos Islands.
I think you have to cut Darwin some slack. He got a lot right, but he worked at a time when what he was thinking was pretty amazing. And so, as you said, Stanley, selection, natural selection, and the whole idea of evolution is important. Not everybody gets it all right. And I'm sure not even Carl got it all right?
I think if you're a scientist, you don't believe anyone ever gets it all right. There's always that next step.
That's right. Well, there's a good saying, science is not about filling buckets. It's about starting fires.
Right.
Right.
Yeah.
All right. I did find the paper with that you published with Carl Stanley. It's called Phylogenetic Placement of the Spyro Somaceae.
Doesn't that sound exciting?
What is a Spyro Somaceae?
Oh, these are these little bacteria that I worked on that curl up. So when you look at them in the microscope, they look like little donuts.
Yeah.
Okay? And so they're different than any other organism that we knew about at that time. There was a small group of them, and they were different enough in many aspects that we wanted to see if, in fact, they were yet another new group.
I see.
But lo and behold, they were bacteria.
So you sequenced the 16S and you were able to place them that way, right?
That's right.
Yeah, very cool. Anything else before we wrap this up? Looks like Elio's not going to make it, but I think we covered it pretty well.
The only thing I want to commend Stanley is I stumbled into episode 66 of the Microbe World video post that you and Curtis Suttle did. So if you're interested in learning more about viruses and oceans, I commend this episode to your consideration. It's 10 minutes, time well spent. And yesterday, as I was introducing our dental students to microbial genetics, I commended this episode to them. I said, just watch this and be amazed, because many of our dental students routinely go surfing, and I pointed out to them the number of viruses in 1 milliliter of seawater as we were going through the story of transformation, transduction, and conjugation, and all wondering why we needed to know about these things. So it's a really neat episode, Stanley. I very much enjoyed it, and you and Curtis Suttle are phenomenal together.
Oh, that's great. Thanks. And there's a second benefit of that, and that is that anything that keeps a few surfers out of the water gives more space for me.
Are you saying it's crowded out there?
Sometimes it is.
Yeah.
But anyway, this was really nice. It was great talking to you guys. Carl Woese was a good friend and somebody I really liked to argue with, and so it was really wonderful to have this chance. And thanks to both of you for everything you guys do, and to the audience. Is such a loyal audience and comes up with really amazing, great questions.
So Carl Woese was 84. He died on December 30th, 2012. And this episode of TWiM, like all the others, will be found at microbeworld.org/TWiM. And if you do like it, you can also find it on iTunes. Go over there and subscribe and leave a comment or a rating, and that helps to keep us prominent on the iTunes directory so more people can find us and learn about this amazing world of microbes. If you have any questions or comments, do send them to us. We love to get them. Today we're going to skip email just in honor of Carl to focus on his life. But do send them to twim@twiv.tv. Dan Malloy is at the University of, San Diego State University. Is that right, Stan?
That's right. Good job.
Thank you so much for doing this with us today, Stan.
It was a pleasure.
It's always
a pleasure.
Always great to have you on. I hope you'll come back not too long from now.
Thank you.
Carl could not have asked for a better eulogist.
That's right. Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
It's always nice that you travel and you still find time to check in with us. We appreciate it.
I try.
You do very well. You don't just try. You do a great job. I'm Vincent Racaniello. You can find me at virology.ws. Many thanks to the American Society for Microbiology for their support of TWiM, and special thanks to Chris Kandian and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins, and you can find more of his work at ronaldjenkins.com. It's used, of course, with his permission. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.